Now we're going to go over some other influences on microbial growth. And one of those, a big one, is gases. Now gases, we've actually talked about this in lab a little bit. So we've talked about the influence of oxygen on the growth of different microbes. And that's going to depend on whether or not those microbes are aerobes, anaerobes, or somewhere in between, like a facultative anaerobe. So we're going to go over that. The two main gases that we really talk about in affecting microbes are going to be oxygen, which we can write as O2, because this is going to be two oxygen atoms that are bound together to create this oxygen doublet. And then we have carbon dioxide as our other gas that's going to influence microbial growth. We're going to be talking most about oxygen in this case, so that's what I want you to focus on when you're reading about this, because that's really going to play a large role in how our microbes actually function. So based on that, I'm removing the carbon dioxide from the board so we can focus on the role of oxygen. Now, oxygen itself isn't harmful to most cells, right? Oxygen is obviously going to be present based on our atmosphere and the gases that are present. So oxygen itself isn't harmful to cells. But whenever oxygen is used and broken down, we do end up with some toxic byproducts. So those byproducts I've written here, these are going to be your toxic byproducts when we talk about oxygen use in cells. So oxygen can lead to singlet oxygen. So as I mentioned before, oxygen is generally present as a doublet in atmospheric gases, but we can have a singlet oxygen form as a product of this oxygen being used in a cell. It can also lead to superoxide ions. So that's shown as O2, but it has a negative charge. So this superoxide ion is going to also be another toxic byproduct. Another toxic byproduct is H2O2, not to be confused with H2O, which is just water. H2O2 is hydrogen peroxide, which you're probably familiar with, probably seen one of these brown bottles before, and it can be used as an antiseptic. Now, that's going to make more sense whenever we go through how these toxic byproducts play a role in cells. So hydrogen peroxide, and finally we have the hydroxyl radicals, which is shown as OH negative. Okay, so let's go over how these are actually processed in cells that are able to use oxygen and still function. So typically what's going to happen with in the presence of oxygen is that you're going to have these superoxide ions that are created, and so we're going to write that down as the O2 minus, and another product that we're going to have in our cells is hydrogen ions, and that's abbreviated H plus. Now these can be converted into H2O2, which is going to be your hydrogen peroxide, and oxygen. Now this is just regular old oxygen, the doublet form that is harmless in the cell. So this is going to be that conversion that can take place if our cells are able to use oxygen. So again, we have our superoxide ion, and we're going to go ahead and balance this equation, which means that we have 2 superoxide and 2 of these hydrogen ions. Okay, so now we have our 2 superoxide ions and our 2 hydrogen ions. Now these can be converted into hydrogen peroxide and harmless oxygen. Now, in order for this to happen, there's an enzyme that is required. And so enzymes, when we talk about enzymes, they typically are what's going to be able to make a reaction take place much more quickly, and that's why they're essential. So sometimes reactions can take place without an enzyme, but they would happen so slowly. You'd be waiting for certain molecules just to bump into each other in the perfect orientation in order for this to take place. An enzyme is actually going to take those molecules, collect them, put them together. It's going to do all of that work. And so that's why we have to have these enzymes functioning in order for cells to actually function. So the enzyme that makes this reaction possible is called superoxide dismutase. So I'm going to write that down as well. And I want you to remember that that is the essential part that's going to make this happen. So we have superoxide dismutase. Now, this isn't too hard of a name to remember because we have the superoxide part. That's actually going to be one of the reactants in this, is this superoxide here. And so we're taking superoxide, and then the dismutase, anytime you see ASE on the end of a protein name, that indicates that it is an enzyme. So anytime you see ACE, remember, that means it's an enzyme, so it's going to be performing some enzymatic function. So we're taking this superoxide and converting it into something else. So superoxide dismutase, that's the enzyme that makes it happen. Now, that's going to be step 1 when we have that superoxide. Step 2 is going to, let me draw a line around this. Step 2 is going to be taking that hydrogen peroxide, because as we mentioned before, hydrogen peroxide is another toxic byproduct in this process. So if we keep that hydrogen peroxide in a cell, that cell is going to die. So we need to convert that hydrogen peroxide into harmless products. So hydrogen peroxide, I'm going to go ahead and balance this equation already. So that's going to be 2H2O2, can be converted into 2 molecules of water, H2O, plus harmless oxygen in the form of a doublet. All right, so this equation, I've already balanced it. We have our 2 hydroxide molecules can be converted into 2 water molecules and an oxygen molecule. Now this is made possible by catalase. So once again, we have to have that enzyme present for this to take place. So catalase, again, we see ASE at the end, which tells us that it's an enzyme. So this means we have to have this superoxide dismutase and catalase in order to convert our superoxide ion into products that are not harmful to the cell. Now, why this matters in terms of microbes is if microbes are in an environment that has oxygen present, they have to be able to convert that oxygen and its byproducts, so the superoxide ion that's going to be harmful, and then eventually that hydrogen peroxide molecule. So in order to convert these harmful byproducts into something that's not going to harm the microbe itself, it needs to have these enzymes. So these enzymes are going to be essential for our aerobes, or obligate aerobes. So you might remember from lab that we have obligate aerobes. Obligate aerobes mean that they have to have oxygen present. And so anything that has to have oxygen present is going to need to have some type of enzymatic process that can convert harmful products into harmless products for that microbe to be able to survive. So that's going to be the obligate aerobes. They need oxygen. And obligate just means they're, like we use obligated, like, oh, I'm obligated to do that, meaning I have to do that. And so in this case, obligate aerobes, that means they have to have oxygen. Now, there's going to be other classifications. So besides obligate aerobes, we also have facultative anaerobes. Now, facultative anaerobes are going to be able to function in the presence of oxygen even though they're technically anaerobes, meaning that they do well under conditions without oxygen. So they don't require oxygen, but they're still able to use it and function in the presence of oxygen. So while they don't require it, they would still have to have some type of enzymatic process that can actually, once again, convert anything harmful into something harmless that's going to be part of the oxygen process. So they don't require it, but they're not harmed by it. Then we also have anaerobes. Now, if something is an obligate anaerobe, this is going to let us know that it cannot function in the presence of oxygen. And this tells us that it doesn't have the machinery, it doesn't have the tools, whatever you want to refer to it as. It doesn't have the enzymes that it would take to convert harmful byproducts of oxygen into something harmless. So if it has superoxide ions present, even if it could convert it in that first step to a hydrogen peroxide is also going to be harmful to that cell. So unless it has the catalase then to convert hydrogen peroxide into water and a doublet oxygen, then it's still going to kill that cell. And so that's why it's going to be essential that obligate anaerobes, because they are obligated, they have to be without oxygen, they need to be in an oxygen-free environment. Because even something like a singlet oxygen would be toxic to them. So they need to be in oxygen-free environment. So I've just made some notes here about the difference between these three types that we've gone over previously in the lab, just to make it clear what the difference is. Now we also have something called aerotolerant anaerobes, and we haven't gone over this part before. Now aerotolerant anaerobes, they're going to differ a little bit from the facultative anaerobes in the fact that they do not use oxygen. So they don't need oxygen. So that's the same as the facultative anaerobes, right? They don't need oxygen either. But with our aerotolerant anaerobes, they don't need oxygen, but they will grow to a limited extent in its presence. So they won't fully grow. They won't grow as well as they potentially could in the absence of oxygen, but the oxygen isn't going to be so harmful that it just kills off the cells completely. So that's the difference between our facultative anaerobe and then our aerotolerant anaerobes. And so you can think about this one because it uses the word tolerant. When we think about tolerance, we're not fully accepting something. We're just tolerating it. We're okay if it's around. It's probably not that exciting for us, right? Like I'm gonna tolerate sitting through this meeting. It's not something that you're really thriving under those conditions. You're probably feeling pretty lousy. And so that's going to be the same idea here. With an aerotolerant, it's only tolerating oxygen. It will grow a little bit, but it's not going to be functioning under its best conditions. So that's what we call the aerotolerant anaerobes. So hopefully that clears up the different types of aerobes and anaerobes and how they're utilizing oxygen and what it really takes to make sure that those oxygen byproducts that are harmful, like our hydrogen peroxide, aren't killing the cells. So that lets us know that hydrogen peroxide is going to be effective in killing something that doesn't have the equipment, those enzymes, to break down this hydrogen peroxide into the harmless water and oxygen. So that means we could definitely use hydrogen peroxide to kill off microbes that are anaerobes. Because anaerobes, the obligate anaerobes, aren't going to be able to defend themselves from this hydrogen peroxide. All right, so hopefully that clears everything up with the different ways that gases can influence our microbial growth.